posed the question, how was the biology of a marsh fish affected by the Deepwater Horizon oil spill? When I mean biology, I'm really going to be focusing on the physiology, the developmental biology, and a little bit on the molecular biology as well. So I'm from Louisiana State, and this is actually a collaborative effort, and I'll get to making the acknowledgement at the end. Now I'm going to keep it simple. I'm going to be focusing on one fish, which is here the Fundulus grandis, or the Gulf killifish. As Dr. Turner alluded to, this is also referred to as the cockerel minnow, or if you go a little east, the bull minnow or the mud minnow. It's a very popular bait fish for those of you that do fish. And my group has been using this quite extensively even prior to the Deepwater Horizon oil spill to essentially monitor environmental stress and essentially look at this as a model understanding the mechanisms of environmental stress tolerance. Now, for some reports, it's the most abundant vertebrate in coastal marshes. Certainly it's very abundant from site to site. You'll find quite a few of them. We use them quite extensively due to the fact that they're relatively easy to raise in captivity. So we can collect them in the field, bring them back to the lab, we can rear them multiple generations. They are also relatively sensitive to chemical toxicity. Now at the end of this presentation, I'll talk a little bit about the fact that this sensitivity will vary depending on the population from which these fish are derived from. We take advantage of the fact that they have transparent embryos, and so we can actually, as the animal is developing through embryogenesis, we can actually monitor inside the animal and looking at how the organs are developing. And we'll see later on is that early development is actually a very critically sensitive time point to oil exposure. Compared to a lot of other fish species, there's a fair bit of characterization of its embryology. We know a lot about its biochemistry and its physiology. Also relatively fair bit about the ecology and the population genetics. We also have a growing genomics toolkit. I'm not going to go into too much of the specifics of some of the more recent techniques like RNA-seq and bioinformatics. However, we do know a fair bit about these nitty-gritty little details, and so we can actually start to exploit some really new age, cutting-edge techniques for using this fish. And so the presentation today is going to focus on predominantly on two studies. This first study by Whitehead et al. Was published in 2012 in Proceedings of the National Academy of Science, and to our understanding it was the first report of a biological impact seen in an aquatic vertebrate. In this second study, my graduate student Devansky, who's now a postdoc at University of North Texas, was published in 2013 looking at really an extension of this study. And so really we're interested in three primary experimental questions. First off, what are the physiological molecular impacts of oil exposure? Predominantly looking at different sensitive stages of sensitivity and trying to address what are some of the fitness level consequences of these oil exposures. And I'll touch on predominantly of this towards the latter part of the presentation. We're not going to go into too much detail about this part. We've looked extensively at the geographical and temporal extent of oiling in the field and try to understand the impacts of different oiling events on Gulf killifish populations. And more recently, we've been interested in understanding how does evolutionary history and exposure history affect the sensitivity to Deepwater Horizon oil. So we all know that Deepwater Horizon oil spill was a very large event. Approximately 800 million liters of oil was released over the course of about 87 days. Oil was spread over a very large area, and this was complicated by the fact that a lot of chemical dispersements were also added, which in itself may help mitigate some of the effects of oil coming in, but certainly increase the toxicity of that oil. This is in comparison a much larger oil spill than the Exxon Valdez, which there's been a lot of characterization of this spill on fish populations, but certainly it's a very large event. These numbers are a little different than what Dr. Turner presented, but needless to say that Louisiana was highly impacted. Over half of the shoreline of Louisiana, about 790 kilometers of shoreline was oiled in Louisiana, and this is about 1,770 kilometers of shoreline throughout the northern Gulf of Mexico. So it's a very large event.
Now, this is actually taken from one of the more heavily impacted sites that we studied. This is in Grande Terre, Louisiana, southern edge of Barataria Bay. Here you can see the fish traps that we commonly use to collect our Gulf killifish. And you can see here at the end of June 2010 that the traps really are on the edge of the oil, and you can see the sheen on the surface. Now, the first study was really, we started getting reports of an oil spill late April, and then it was certainly before my 40th birthday became apparent, well, this is a pretty big deal. So we started getting all our field collection equipment, and we first off started going east because all of the models suggested that oil was going to actually impact predominantly Mississippi and Alabama. And so we did a lot of sampling, or we did some sampling in Alabama at 3 sites, Fort Morgan, Alabama, Upper Mobile Bay, and Bayou La Batre. We also did sampling in 2 sample sites in Mississippi, Bay St. Louis just east of here, and Bellefontaine Point, and our 1 site in Grande Terre, Louisiana. Now, by the time we started sampling Grande Terre, this started becoming like Fort Knox, and so sampling actually became relatively difficult, but we managed to get onto some of these sites. Now, we basically, in a nutshell, just to kind of summarize a lot of chemistry data and a lot of remote sensing data, of these 6 different sites, based on our sampling, Grande Terre was the only location that essentially was impacted significantly by oil. And we say that it was the only site that had significant elevation of oil in the sediments and in the water. Okay? It also had, in comparison to these, in which oil came close but never reached within the time frame, at least until August 2010, were not impacted by oil. Most of the polyaromatic hydrocarbons were sequestered within the sediments. We had extremely high levels of PAHs in the sediment. And in fact, as I showed previously, this is what our marsh edge looked like in Grande Terre, Louisiana, heavily impacted. Here you can see the boom. And you can see here that these are Gulf killifish, that they were in fact exposed. Now, the first study was not funded initially, and so we went out there and tried to do what we could within the context of not having, certainly the uncertainty of knowing how we were going to pay for it. But finally we got some funding to actually measure some of the things. But ultimately we're interested in measuring tissue-specific responses to these fish collected within the field. My group was predominantly looking at physiology and protein level expression changes, and in collaboration with Andrew Whitehead's group, looking at genome-wide mRNA transcript changes. So basically, just in a 5-second biology course here, mRNA is basically the blueprint from which proteins are made. So it's that intermediary step between the gene at the DNA level and the protein. And so we're going to be looking extensively at mRNA transcript levels, and I'll just very briefly go through that when I show them. We also have been doing a lot of work on early life exposures, a lot to field collected waters and sediments. More recently, we've been doing a lot of work on various different manipulations of oil and water spiked with oil and sediment spiked with oil. We've been looking at various measures of development, time to hatch, hatching success, and gene expression, as well as physiology. Now, this is what a microarray looks like, and this is actually starting to replace more recently with newer technologies like RNA-seq. But essentially, each one of these dots represents a specific or unique mRNA transcript. Okay, so this dot here may represent a mRNA transcript that produces a certain protein. Okay, and what we're able to do is, based on the level or the color that's indicated, have an indication of whether that mRNA has increased in abundance, remains constant, or has decreased in abundance. And so there's approximately 6,800 different transcripts, and so this is a genome-wide in as much as we're able to look at a good assortment of the different types of mRNA transcripts that would be in the tissue. And all of these spots are essentially, would fit within that little spot. So essentially use robotics to put these little specks of these probes on there, and then we essentially hybridize it with actual mRNA collected from the tissue. So that's, in a nutshell, the technique. And so what we're able to do is generate various scans that look like this. And in fact, to our understanding, this was the first application of this technology to oil contaminated fish in the field. And so let me just very quickly orient you to this. Here, each one of these blocks represents our different populations going from west to east. So Grant, Terre, and Louisiana population is always on the far left. Each one of these different columns, you'll see that for most there's 3 different columns. This would be May 2010 before oil hit. This would be the end of June after oiling. And this was in August 2010 when there started to be somewhat of a recovery, when certainly a lot of the visible oil on the surface was starting to dissipate, but certainly there was a lot of oil still in the sediment. And so each one of these columns represents the same timing from pre-oil to sometime after oil and recovery. Now, yellow represents a very high upregulation in that abundance of that mRNA transcript, whereas blue or magenta represents a large downregulation. Now, in a nutshell, what we find here is that the Grand Terre, coincident with the arrival of oil, which at this site, and I forgot to point out, oil arrived at this site around June 10th to June 14th, okay, based on some of the satellite imagery that we had obtained. This sampling was done in around the end of June. So in around 2 weeks after the initial oiling of this site, those fish that we had collected, we had taken the livers, and what we found, as shown here by this high yellow, is a group of mRNA transcripts that were very highly upregulated, and another group of transcripts that were very highly downregulated. Now, it looks like a mess here, but essentially this site here is, in the fish from those sites, were the only ones to show this pattern. Okay? Now, these groups of transcripts are what are called AHR ligand regulated, aerial hydrocarbon receptor ligand regulated mRNAs. So essentially, and I'll go on to talk about this, what these different transcripts represent, but essentially these gene expression in the livers of GT fish are, the differential expression is coincident with the location and the timing of the arrival of oil. Now, just in a nutshell here, this polyaromatic hydrocarbons, when they enter into a cell, they bind to this AHR receptor. When that happens, these chaperones come off and allows the ligand together with the receptor to go into the nucleus, where they bind to this other fellow here, ARNT, and they can bind to xenobiotic response elements that are just upstream of many different genes. When this happens, whatever that gene is, it will produce, increase the abundance of nuclear mRNA. This nuclear mRNA is what we're basically seeing here. Okay, so we see a whole bunch that are upregulated in response to this, or a whole bunch that are downregulated. We say that they're AHR receptor ligand mediated because they all follow through, or all basically follow through this pathway. One of these is cytochrome P450 or CYP1A, which is involved in the metabolism of oil. So essentially, you've got this hydrophobic substance. When it enters into the organism, the cells try to make it more water soluble in order to metabolize it. Now, this is a good thing. You're trying to metabolize the oil. But there's a lot of indications that upregulation of this pathway, in fact, leads to some of the downstream cardiovascular and developmental effects that we often see in embryogenesis, during embryogenesis. So although this is a good thing in trying to metabolize the oil, it's also been shown that if you increase it too much, that it actually has these developmental impacts that I'll show momentarily. Now, we also see here that cytochrome P450, if we look at the gills, from our gills from fish collected in May 2010 versus those that came immediately after oiling, okay, and during the recovery period, the initial recovery period, we see cytochrome P450 is shown by that dark staining pattern. It's increased in abundance. What we're looking at here is immunohistochemistry, where we basically take an antibody against that protein, and we're able then to look at not the mRNA, but rather the protein, and to see if it's actually being upregulated. And so it's suggesting that these animals are in fact changing. Now, I want to clarify, this is not a mutation. I've had US congressmen that try to tell me that this was a mutation. I remember I had a Russian reporter come into my lab and says, "I'm here to see mutations." And I just go, sorry to say, I have no mutations to show you. These are actually protein level effects and are not mutations. Okay? Now, in fact, we looked at as late as August 2011, we see that there's still, despite the fact that oil started to decrease in concentration in the field, we still see a very high level of CYP1A expression in the gills. Okay. If you actually look at the gills, there is actually a lot of damage. There's a lot of hyperplasia, and there's a lot of mucocytes. It's a little hard to see on this image, but there is some physical damage. Not a mutation, but rather physical damage to that gill epithelium. And I should say that the gill really is, it performs a lot of function. It actually functions much like our lungs in gas exchange. It acts like our kidneys in ion and acid-base regulation. So it does a lot of different things. And so, essentially, in a nutshell, we show that these animals, in fact, have been exposed to the toxic components of oil. Okay, so PAH is essentially getting into the animal, and though the oil in the hydrocarbons may not be accumulated in the tissues to a great extent, they are getting into the animal and are exerting a biological effect. Now, we also found a lot of gene transcripts that in the Grand Terre population decreased substantially in their abundance in the Grand Terre. You see here in these other populations where you see yellow, they're increasing. So these animals, as you're going from May through June into August, these animals are starting to reproduce. This is their reproductive cycle. And so that oiling event really coincided with the peak of reproduction for this species, which typically happens from around May through about September and October. Okay? There's a downregulation of a whole suite of different genes involved with reproduction, zona pellucida and choriogenin, which are involved in the outer eggshell, if you may, the chorion of the egg, vitellogenin, and so these are downregulated in a Grand Terre fish. We also found a very high upregulation, as shown by this high yellow comparison to the scatter, the noise here seen in the other populations in various genes associated with proteolysis. What's interesting is a paper came out around the same time showing that this AHR receptor, here they call it the dioxin receptor because it actually binds dioxins as well. This is the equivalent of the AHR receptor, is a ligand-dependent ubiquitin ligase. So basically going through this schematic here, PAHs can activate the AHR receptor. They can act as this ubiquitin ligase. It can increase the activity of ubiquitin ligase activity. And this activity can actually degrade sex steroid receptors. And so it's a mechanism for actually degrading receptors. And so we propose that this in itself could, in fact, suppress sex hormone-induced gene expression in these animals. And we propose, although we didn't have at the point endpoints to really confirm it, that there's indications that reproductive fitness could be compromised if you have PAHs high enough to essentially elicit the HR pathway, which in fact we showed to happen. So there is some suggestion of compromised reproduction, essentially altered estrogen signaling. Now, we've done a lot of work in cardiovascular fitness, and I'll spend a lot of time from now on talking about this, but there's also a lot of gene transcripts associated with cardiovascular physiology that were also differentially expressed in our Grand Terre population that were not seen in our other sites that did not receive oil. And so there's some indications of altered cardiac physiology. Now there's several studies that have come out, various consortium and actually independent studies. This is a study by Cardona's group where I highlighted a few points here. We show that environmentally realistic exposures to pHs cause specific dose-dependent defects in cardiac function. In this case, they were looking at different species that I'm just showing you, the bluefin tuna here, but it's essentially showing that cardiac function has been compromised and it culminates into pericardial edema, which is swelling around the heart and other secondary malformations. And suggests a considerable portion of the Gulf waters collected during the oil spill had pH concentrations above this threshold. It suggests that this is not just an academic exercise, but in reality that a lot of the fish populations in the Gulf at some point would have been exposed to concentrations that were high enough to elicit these cardiovascular impairments. And cardiovascular impairment, as you might expect, if your tinker's not working very well, it's going to be a little difficult to do a lot of things, okay? And so they found that even subtle changes in cardiac function during exposure during embryos actually had long-lasting effects. And so animals, when they became adults, and this was actually looking at zebrafish, but regardless, all vertebrates, their cardiovascular development is very similar, that these animals had a hard time swimming. And so there is clearly there's this cause and effect. There's some real world problems that the animal has to deal with. It has to swim to collect its food and to evade predators. So in conclusion to this part, there was a large signal of hydrocarbon exposure that existed in Louisiana marsh fish collected in situ. We saw that AHR pathway was upregulated, and certainly we saw CYP1A protein highly upregulated in the gills and livers from these fish. And the transcripts suggest potential for reduced reproductive capacity and alterations in cardiovascular physiology. Now, kind of moving along quickly in here, this other study actually was kind of piggybacked on some of the samples that we had collected, but also took advantage of some additional sampling we did in 2011 and 2012. Basically, we did a lot of sampling, and I'm only going to focus in on a few sites. Some of these were reference sites, some of these were oiled sites, as Dr. Turner alluded to. The oiling was very patchy, but we had sites that were, as MBS here, had very low pHs in the sediment, and others that had Wilkinson Bay had moderate oiling in the sediments, and Grand Terre had very high levels in the sediment in 2010 and reduced in 2011. Here's my graduate student at the time collecting sediment cores from the Marsh edge. We basically took these sediments and we developed these mesocosm studies where we essentially took sediment and we overlaid it with 1/3 strength seawater, clean seawater. These are Teflon baskets that we basically would put our fish embryos so they would never sit immersed directly into the sediment. And so essentially these animals were in the overlying water, overlying the sediment. In a nutshell, we basically here found that heart rate, which is a pretty good indicator of cardiovascular function, was significantly reduced by about 30 to 35%, which is actually a very significant reduction. So in the Grand Terre sites, 2010, there was a very large, there was slightly attenuated reduction in heart rate in the Grand Terre sediments collected in 2011, and comparison to these other two sediment sites. So it seems to follow the dose response curve. We looked at percentage hatch, and what we find here similarly is that when we expose the embryos to sediments collected from Grand Terre in 2010 and 2011, there was a very significant reduction in the ability of these embryos to go on to survive and to develop and eventually to hatch. Okay? Now, if we actually look at this, a more nuanced look at this, what we find here in red, these are the percentages of hatched and these are unhatched. So what we found is when we expose these animals to very heavily contaminated sites, the animals remain within the chorion, and here's the embryo inside its little eggshell there. It's still alive, it's not doing very well, and it never actually hatches. Essentially, it doesn't have enough vigor to actually escape its chorion. And so even though it's still alive, and really for all intents and purposes, it's dead. And so we hear a reduction in here. And if we basically, and we also see from these, within these embryos, is that we start to see a lot of alterations in the proper folding of the heart. So when we expose embryos to high contaminated sediments, what we start to see is the normal folding pattern, which shows this kind of L pattern of folding between the atria and the ventricle, tends to become straight. And so the heart is essentially not folding like it would. And so there's some alteration in the developmental process that allows it to fold that's not happening. As a result, the heart is not functioning. We start to see a lot of edemas around the heart. It's a little hard to see here. We see craniofacial abnormalities. We see a lot of incidences of hemorrhage in the animals as well. In fact, when we look at laboratory spike sediments, and here we're looking at total pH, we see this very nice correlation between the amount of polyaromatic hydrocarbons in spiked sediments and percent hatch, and concentration of pHs in the sediment and heart rate. You never get much below about a 30 to 40% reduction in heart rate, but these animals are essentially doomed for failure. Okay, they will essentially not develop, and as you can tell, they do not hatch at these very high levels. And in fact, when we took laboratory spiked sediment experiments, so we just essentially took the oil and mixed it with the sediments, this almost fits exactly with when we took the sediments from the field, brought them in the lab, measured the pH concentrations, and developed similar concentration curves. And so exposure to field collected sediments increases time to hatch and reduces the percent hatch of killifish embryos. And we have these teratogenic, or these developmental abnormalities in those animals, and certainly affected cardiovascular fitness. Now, what's next? Well, my group has been funded over the last couple of years by the National Science Foundation and the National Institutes of Environmental Health Sciences. Somehow we managed to convince them that a killifish is a vertebrate, and humans are vertebrates, and so we can use a killifish to study human health. This is actually a collaboration with two other folks, Chris Green from the LSU Ag Center and Andrew Whitehead, who's now at UC Davis, long-term collaborators of mine. And essentially, we're interested in this early life development, how exposure during early life development influence juvenile and adult performance, and also exposure to other environmental stressors, and how those are compromised by early life exposure to oil, and ultimately the effects on reproduction, and ultimately that reproduction and what influences it has on early life development in the next generation. So we basically oil embryos, and we look at those animals during development. Okay, so looking at critical developmental stages and impacts on later life, adult exposures to oil and the impacts on reproduction, adult and early life exposures and those transgenerational effects, and so what effects eventually make it out to F2 and F3 generations. And so there's a lot of epigenetic effects that are not genetically inheritable, but things like methylation of DNA or histomodifications that actually can transmit effects across multiple generations. We're interested in mechanisms, and so I'm a physiologist, and so we're ultimately interested in understanding some of the underlying mechanisms. And one thing that I'm going to present a little bit of today is genetic variation and exposure history. And so the ability that this may either protect or sensitize individuals to oil exposure. So very quickly, what I'd like to do is talk about some unpublished work that we're currently working on, where we're basically collecting different populations of killifish from Florida through Texas. Okay, 3 of these, as shown in blue, are reference population, St. Teresa, Florida, which I'm not going to talk about at all anymore. A population that was derived from southern Louisiana, which we actually have maintained in culture at LSU Aquaculture Facility, and Gangs Bayou, which is near Galveston, Texas. So these don't have any known history of oil or oiling or hydrocarbon exposure. This is in contrast to these shown in pink, Grand Terre, Louisiana, collected in 2015, which we know is being oiled, and another population collected in Vince Bayou, Texas, which has a long history of hydrocarbon exposure. This is a U.S. Superfund site, and in fact, there's data from Cole Matson's group suggesting that this population has become adapted, so it can actually tolerate orders of magnitude higher concentrations of hydrocarbons that would normally kill this species of fish. Okay. So we basically use these, and I'm just going to focus on the 2 Louisiana and the 2 Texas populations. We've brought these into the lab and use this as a brood stock. And what we're able to do is take males and females, parental controls, they've never seen oil, and we can fertilize them and get embryos. We've also exposed parents, the males and females, to a WAF concentration, an oil to water concentration for at least 40 days to about 50 ppb of total polyaromatic hydrocarbons. And we once again fertilize and get the embryos. Interestingly, what we find is that when we look at fertilization success, okay, if we look at the parental populations that we collect from the field, but we don't expose them to oil, excellent fertilization. About 95% using our in vitro fertilization method, 95% of the eggs can be fertilized when we expose them to the sperm. Okay? Interestingly, when we expose these parents to oil for 40 days, look at these populations from Louisiana and the Ganges Bayou. So these are reference population that's exposed to Deepwater Horizon oil. Fertilization drops to 25%. All except this Vince Bayou, which comes from this highly contaminated site. When we expose those parents to oil, we still get excellent fertilization success. We now look at the embryos and expose them really as a stress test to look at exposing them to 0 to 56% WAF, 100% being 2 grams per liter of a high energy WAF that we created. And we basically, if we look at Ganges Bayou, the parental control and the parental exposed, we see a significant reduction in heart rate as we increase oil, like we would expect, and as I showed previously, but no difference really between the parental control and the parental exposed. Our Vince Bayou, which is our population collected from that highly contaminated site, the heart rate is really refractory. We can expose them to very high levels of oil, no effect on heart rate, either in the parental controls or the parental exposed. Interestingly, when we take the Louisiana population, slightly higher heart rate in the parental control, but they seem to be more sensitive. We're actually seeing a significant reduction in heart rate, both in the parental controls and the parental exposed embryos. Embryos collected from parentally exposed animals. We see a significant reduction, and this reduction in heart rate is actually greater than we found in the Texas population. When we look at the Grand Terre, the embryos collected from the parental control parents, once again, like the Louisiana, our aquaculture population, they're also very sensitive. Notice that I have no pink bars. Pink bars is because those individuals that did get fertilized, they didn't survive. They didn't survive even when they weren't exposed subsequently to oil. And so there's something about this Grand Terre population. When we subsequently expose them to oil, expose the parents to oil, the embryos, even those that survive and are fertilized, don't go on to survive. This is at 4 days post-exposure. And so, conclusion, genetic variation in exposure history can either protect or sensitize individuals to subsequent oil exposure. So it's something that we really need to get a handle on, is really understanding population variation and the sensitivity. There's a lot of information here, but F1 embryos from Louisiana populations appear to be more sensitive than those collected from Texas. The F0 adults, the adults from the adapted population are recalcitrant to Deepwater Horizon oil. In other words, they retain high reproductive capacity. And similarly, F1 embryos from these adapted populations is more resistant to cardiovascular teratogenesis, or these developmental effects. So I'd like to thank the Whitehead group, the Green Lab, and just a few of the members of the Galvez Lab. Really, we're a pretty large operation, and it could take a whole page here to fill those in, but I have a good group of undergraduates and graduate students that keep the fort running when I'm not there. Okay, thank you very much.
Okay, in the interest of time, can we limit it to 2 questions, and then we're going to have lunch, and at that time you can attack Vern with all the extra questions you have that you're holding back.
Yes.
So, you touched on this, but how long does the Cipro 1A or things like that last once it's exposed? I mean, you have an expression, and is it sensitized, trained, does it recover with no history? How long does it last? Is it just that generation, or is it carried forward?
You know, there's some indications that, we talk about these epigenetic effects, and so these are things that you may see in the news. What happens when your mothers are exposed to some contaminant, and what are the downstream effects? There appears to be some indication that that there is this epigenetic effect where there's these modifications that happen around the genes associated with this AHR pathway that, these include methylation of regions of the DNA or histomodifications, and it actually has long-lasting transgenerational effects. And in fish, we don't know. It's really, there's been no follow-up on that pathway. There's a group at Woods Hole, Mark Hahn, who is currently looking at that, and we're actually interested in following up on some of these pathways as well. But there hasn't been too much work.
Is it reasonable?
Excuse me?
Is it reasonable to carry forward or not?
Everything's reasonable if you have a budget. If you have a budget and you have the manpower, yeah, but certainly. So to get your effect, in our populations, we really haven't followed on the washout where we've exposed animals and then looked at the time course washout. In other words, reduction in the CYP1A expression. All of these have been collected. So when we go in the field in 2011, 2012, these animals have a very, very large CYP1A expression in the gills, in the liver, in the kidneys, and in the intestine. The intestine these are very high expression. So these animals, marine fish drink, unlike freshwater fish. Marine fish drink, and so there's likely some ingestion of water, but also particulate matter, and certainly through the food where it might. But these animals are showing expression, and this is in the face of reduced pH concentrations in the field. So you would expect that these signals are relatively long-lived.
Another question for Dr. Galvez? Okay, it's a hungry crowd, right? All right, thank you again.
Thank you.